All right, we are back talking about the cell cycle. I feel like we've talked about this a couple of times because we've been covering it in the lab. We'll go over it one more time. So this is how we diagram the cell cycle or the life cycle of a cell. And you want to think of it starting right there. So M represents mitosis, which we probably know by now is cell division. So imagine one cell has divided and becomes two. The two start in G1. The G stands for gap phase. As it says here, this is when growth and regular metabolism of the cell takes place. Everything from G1 through S to G2, everything that's not M, is referred to as interphase. So if a cell decides it is going to divide, it then enters S phase, which is the synthesis phase. That's when DNA is replicated. And then it goes through another gap phase and before entering or committing to mitosis. Those cells that are not going to enter mitosis, certain cells like neurons and muscle cells rarely or never enter mitosis. So they stay suspended in the form of G1 called G0 or G0. So as this says here, right, this is a checkpoint before entering S phase. The cell has to make a decision. Is it going to prepare to divide or not? And then you have another checkpoint here at the end of G2, right? In addition to, as it says here, growth and preparation for mitosis, that preparation includes proofreading the DNA and making sure that everything in the cell is prepared for a successful replication so that you don't replicate when you have mutations in your DNA and then pass the mutations on to your progeny, which are then going to have them and pass them on to their progeny. So you are going to see when we talk about our cancer case study that we refer to genes that act like accelerators and genes that act like brakes. This is where the accelerator genes would be acting, saying, yes, let's do it. Let's go through S phase, let's enter mitosis, let's divide. And then you have another set of genes where this is where you would apply the brakes, usually. No, it's not a good idea to divide now. Brakes can work here too, but that's kind of how I think of it. So now we have to talk about replication, what happens during S phase. Again, kind of like cell membrane, this is a somewhat complicated process. You do not need to be able to explain it. I like to cover it, as I like to do, in a certain amount of detail for those of you that want to understand it. But for those of you that just can't quite yet wrap your head around the mechanics of it, don't worry, I will tell you the key points that you need to know. So first up, replication, what we're talking about is using DNA to make more DNA. So here you've got one chromosome, you're going to pull it apart, replicate it, and end up with two chromosomes. So the first thing you want to know is that there's an enzyme that operates right there called helicase that unwinds the DNA. So it doesn't just separate the two strands, but it has to spin and untangle the DNA, because you can imagine if you were just pulling apart those two strands, all of these twists behind it would get tighter and tighter and tighter. And the helicase has to deal with that. So it unwinds the DNA. Then you have DNA polymerase, which is what manufactures a new complementary strand opposite your two pre-existing strands. So that part you need to know. Helicase, polymerase, one double-stranded DNA molecule gets turned into two double-stranded DNA molecules. Some of the other details that are not as important is that you will notice these two polymerases are working in opposite directions, because this one can work in the direction in which the DNA is unwinding, it can polymerize continuously. It is called the leading strand. The lagging strand works in the opposite direction, so it'll start here, make it out to the last place it started, and then let go and come back to here and work to there. So it's always behind. It's called the lagging strand. The reason why, for those of you that like to know, is that you need to remember that your two strands have directionality to them, and the way they're put together, they are oriented opposite one another. So on this strand here, you've got your sugar with the phosphate sticking out. So it's the phosphate end of the nucleotide that is exposed. And then over here, it's the sugar side. So you've got, phosphate, sugar, phosphate, sugar, phosphate, sugar, you end with sugar. Here is phosphate, sugar, phosphate, sugar, phosphate, you end with phosphate. And this phosphate is part of, or needed for, the catalysis of adding another nucleotide. So you can only add to this end of this strand, and you can only add to this end of this strand. So if this were a piece of DNA, right, let's imagine this piece of DNA is being replicated. So I'm going to do this here. Whoops, they're in the wrong direction. They should be sticking out that way. So imagine this little section of DNA here, double-stranded up until this part, and then your single-stranded is like this part of the DNA here. Double-stranded till you get to here, then it's single-stranded. So on this end, you can add nucleotides here, and that would be the way your leading strand works. And then when you're replicating opposite this strand, right, you have to add here, because there's your phosphate. So this phosphate here would be located right here on this strand. Way more detail than you need to know, but it's fun to understand these things. And then I am going to play this very brief video for you. It's maybe a minute, but I have to, it won't screencast in presentation form, so I have to go to the browser. This is the same video actually in YouTube, and you should get, hopefully you get the computer audio. Hold on.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied. Using computer animation based on the latest
So
research,
this is
we are now
cartoon
able to see how
animation
DNA is
from
actually copied in living cells.
high-resolution molecular imaging that is done with techniques I don't fully understand. But you can be relatively confident that this is a reasonable facsimile of exactly what replication would look like if we had a camera that was good enough to take pictures at the molecular level like this. So this does represent to a fair degree of accuracy what it actually looks like inside of your cells.
You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase.
Right, so this is the one
Spins the
strand
DNA
that's
as
being
fast as a
unwound,
jet
and this
engine
is
as
the
it
other
unwinds
strand.
the double
We're going to see
helix
this
into
one
two strands.
is our leading strand, and this one is our lagging strand.
One strand is called continuous.
This one.
Off to the right. One strand is copied
This
continuously
polymerase can
and can be
move
seen
or can
spooling off to the right.
enzymatically add in this
Things are not-
direction. So as the DNA is being spooled off in that direction, the polymerase can work in that direction. And then on this side, the polymerase has to work in the opposite direction So it's always trying to play catch up, kind
So
of.
simple for the other strand, because it must be copied backwards. It is drawn out repeatedly. Not so simple for the other strand, because it must be copied backwards. It is drawn out repeatedly in loops.
And see,
and
this
copied
is one section that it copied out, and it started
The end
somewhere
result
back here and was
is
copying,
two
copying, copying,
new-
copying, and then when it gets here, it's going to let go, reach back over and pick up this single strand here and keep going until it meets up with wherever back here is the end, the beginning of the last segment, really. So we'll let that play for a second. And there it goes, pulls out a new strand. Okay, and if you want to know even more about it, you can watch our friend Hank Green talk all about DNA structure and replication in even more detail than I did. So far we are knowing that DNA replication is taking one DNA molecule, turning it into two DNA molecules. It is helicase that unwinds our double-stranded DNA, and DNA polymerase that polymerizes or manufactures two new complementary strands opposite our two original strands. So you're going to start off with your double-stranded DNA. Let me pick a different color and we'll put our bases in. Right? The artistic ability is just amazing. Then what we're going to do is we're going to melt that. So there's one strand, there's the other strand, and then we'll put, these are the bases. Because remember, there's two complementary bases facing each other there. So let's say this is A, G, C, T. Then polymerase is going to come along and put a new strand here, just using the rules of complementarity. So does anybody recall what's supposed to be across from A? If you're thinking T, you are right. Across from G. If you're thinking C, you are right. And then, of course, G is across from C, A is across from T. And I don't think I had the time to actually explain this as well as I would like to, because we didn't want to get too deep into RNA and DNA structure. But, oh, we don't have a good enough figure for it. Oh no, this kind of does it justice. So there are two things you want to note about how the complementary base pairs work. One is that you always have a double ring base across from a single ring. So double, single, double, single, double, single, single, double, single, double. So what this means is that there's always a uniform distance in between your two phosphate sugar backbones. So there's an active site in the polymerase where this reaction takes place. If you try to put a double-ringed DNA containing, or a double-ringed nitrogenous base in a nucleotide opposite another double-ringed one. So if you tried to put guanine here opposite adenine, G across from A, they're too big, they just wouldn't fit. And then when you do get the ones that fit, they form hydrogen bonds. So at the same time, if you tried to put, let's say, this blue, I don't know what the blue is, cytosine, here across from your thymine, C across from T, it would be too short, and it wouldn't be able to bridge the gap between this side of the molecule and that side of the molecule. It wouldn't be able to hydrogen bond, in part because of its size and in part because its charges are in different places. Here you see these two nitrogenous bases form two hydrogen bonds between each other. These two form three hydrogen bonds. So when you have the two proper bases across from each other in the DNA structure, they fit physically together. They are the right size and shape, and they also have the right distribution of charges so that they can properly hydrogen bond. So if we go back to that cytosine, and we tried to put a smaller wrong base in there, it wouldn't fit properly. It would fit too loose in the active site, and it also wouldn't stick in there very well because it's not able to hydrogen bond. So the proper nucleotide would just bump into it and knock it out of the way, because the proper nucleotide is going to form those hydrogen bonds. It's going to stick in the active site of the polymerase, claim that spot as its own, and then be added to the growing strand. It's a little bit more detail than you need to know, but if you understand these mechanical details of DNA replication, then you can see it is a very mechanical process. So the complementary strands are synthesized in the proper order, because if you try to put the wrong bases across from each other, they simply do not fit at a mechanical level. So there's no hocus pocus, special forces at play. It's just mechanical forces and being able to hydrogen bond that determines the structure of the double helix and how to replicate it. So going back to this, because I'm making this very long, this is our original double helix with the 2 phosphate sugar backbones in blue. We added a new phosphate sugar backbone with the complementary bases in green here, and then we'll end up doing the same here. And yeah, I drew it in the same direction, but it should be the opposite direction. So then what you end up with is 2 new chromosomes, each of which contain 1/2 of the original molecule. So 1/2 of your original chromosome still exists physically intact in your 2 new chromosomes. That's why this is called semi-conservative replication, because roughly half of the molecule is conserved every time you replicate it. And I know I've thrown a lot of new terminology out at you, but once you understand the terminology and get comfortable trying to imagine the DNA structure, polymerizing a new strand becomes a simple mechanical process. That last step is very easy to understand once you get all of the structure and hydrogen bonding patterns straight in your head. So then we go to gene expression. This is totally new but related, as it says right here. So in our first part of this screencast, we were just talking about making copies of DNA. Now we're actually going to be using DNA for the reason that it exists. So we're still going to be starting with DNA, and things are going to look similar, but it is a totally different process. This is called gene expression. So I've probably already said that the information needed to encode proteins is found in your DNA. Or more specifically, the information needed to encode a polypeptide, right? 1 strand of amino acids is encoded in your DNA. So one portion of your DNA that includes the code for 1 polypeptide is a gene. So when we talk about using the information stored in DNA to make proteins, that is called gene expression. And this is what your cells do throughout interphase, and it is the difference between a muscle cell or a neuron or an epithelial cell or a secretory cell in your sweat glands. All of your cells start out with the same DNA, but what makes one cell different from another is the genes that it expresses. This is the heart and soul of development and differentiation, how one single fertilized egg can develop into the 200 or so different cell types that you find in your adult body. All right, so how does it work? Just to review the code, right? This is your genetic code, and the code is in the bases. It is not in the phosphates and the sugars. So these alternating A's, G's, C's, and T's contain the information needed to put together the amino acids. So cysteine, methionine, leucine, alanine, histidine, valine, I can do all of them. But you don't want to hear it. So that's your genetic code. So what we're going to talk about then is how is this decoded? Mechanically, how is this used to assemble that? Because it is all mechanical. Everything works based on things that are mechanically designed to stick to one another. So we are going to have a two-step process to express a gene. First up is transcription, which is going to take place in the nucleus, and then next is translation, which is going to take place in the cytoplasm. And yes, I know I changed the slides. I can't help myself when I see a way to make it better. So this is a little bit different than what you have, but it doesn't contain anything that you don't have. So here is transcription, which is taking place inside the cytoplasm. And you're taking DNA and using it to make mRNA. And then in the cytoplasm, you're using the mRNA to assemble a series of amino acids and make a peptide. The way I think about it is for transcription, I think of a scribe, a religious scholar born before the days of the printing press, or maybe not even a scholar, just a lowly acolyte, trainee, whatever, that their job was to copy out copies of whatever, the Torah, the Bible, the Quran. So there would be multiple copies of these texts. And so all they were doing was making copies. They were transcribing the same material again and again and again. If you are going to go from one language to another, like the old Aramaic into Greek and then Greek into English, that is translation. So here you're going from bases to bases, right? DNA bases, RNA bases, you're just transcribing the same information in the same language. Here you're going from the language of base pairs, or bases, to the language of amino acids. So you're translating into a different physical language. So step one, transcription. This, in a nutshell, means you are using DNA as a template to synthesize what we call mRNA, or messenger RNA. So the way it works is one portion of your DNA double helix is pulled apart or melted. Another enzyme called RNA polymerase gloms onto what is called the anti-coding strand of the DNA, and it manufactures, as it sort of makes its way down the DNA strand, a single-stranded mRNA transcript that is complementary to the DNA. So remember, U gets substituted for T in RNA. So if there's an A here, there's a U there, T, A, G, C, A, U, C, G, the whole way down. Right? So the same rules apply. If you try and put anything but U across from A, it's not going to fit, it's not going to hydrogen bond, and it's going to get knocked out of the active site by the proper amino acid, which will fit and will hydrogen bond. So it's the same base pairing rules that work on the same mechanics to ensure that this mRNA is always complementary to that DNA. And then this is going to get exported to the cytoplasm. This is where things get complicated, but still very much mechanical. So this is translation. Now the mRNA is going to be read off and translated into amino acids. You don't need to be able to describe the whole structure, but you do want to know what translation means, which is what I just said, using mRNA to assemble amino acids. and then you want to know what the ribosome does, what the mRNA does, what is a codon, what the tRNA does, and what is an anticodon. So the first thing we're going to talk about is the mRNA and the codons. And I've changed these slides a little bit too, but it's still all the same material, so bear with me. When the mRNA gets shipped out to the cytoplasm, the ribosome always finds the same first 3 letters of the code. This is called the start codon. So if you look here, there's some upstream untranslated mRNA. This mRNA is not used to make proteins. It is used to help the ribosome physically grab hold of the mRNA and figure out where it's supposed to start. So the first section of the mRNA that the ribosome starts to read off is called the start codon, and it is always AUG. This is how the ribosome, or part of how the ribosome, knows where it's supposed to start translating the RNA. And then it is going to move along the mRNA and read it off 3 bases at a time. Like, they don't really have it drawn here, but there are 3 different active sites, SP and something else I forget, and each of the 3 active sites is just big enough to fit across 3 bases in the mRNA, and that's the physical basis for this 3-letter codon. So the translation is going to take place in 3-letter words on the mRNA called codons, and each codon is going to call for, as it says down here, a specific amino acid. It is the transfer RNA that physically bridges the gap between codons and amino acids. So it is actually like the decoder ring that decodes the code to deliver the proper amino acid. And this I think I've also changed a little bit too. So this is what a tRNA looks like in great detail. It is one long single-stranded RNA molecule that folds back upon itself. So it's got these double-stranded regions here because there are regions of complementarity within this long molecule that allow it to hydrogen bond and take on this sort of cross-like shape. On one end of the tRNA are 3 bases that are going to fit into this active site right here, or these active sites. Those 3 bases are sticking out, not sticking in like these bases here are, where they're hydrogen bonded to each other. They are called the anticodon. And the way amino acids are made is that each anticodon is always linked to the same amino acid. So if we go ahead here, right, every time the tRNA has the anticodon UAC, then there is an enzyme that is going to recognize that and always add methionine. So this right here is what is bridging the gap between bases and amino acids. So then what's going to happen is that this tRNA with this anticodon is going to stick here because this anticodon is complementary to the AUG. So here you have AUG, and if you look down here, that puts A and U across from each other, U and A across from each other, and G and C. So the same rules of base pair complementarity dictate which anticodon is going to show up and stick across or next to each codon. So every time AUG shows up in this mRNA sequence, there is a UAC tRNA that is going to deliver methionine. And here is UUU. The anticodon for that is AAA. So every tRNA with the AAA anticodon is always going to have this amino acid, phenylalanine, attached to the other end. So that every time UUU shows up in the mRNA as the codon, the tRNA is going to deliver phenylalanine because that is always matched up to the anticodon. So you'll notice now you're moving along. The AUG was there. Now the AUG is a little bit further. Oh, they did it wrong. This should be moving back, and this AUG should be right there, and that AAA should still be stuck to that. So the ribosome is going to move in this direction, and then the growing polypeptide strand keeps getting added to the new arrival amino acid. So then phenylalanine is going to get stuck onto arginine, and arginine is going to get stuck onto the next amino acid, which would come in for... Oh, they've got them all messed up here, because this GCU should be across this CGA. But that's how it works. It's codons have matching complementary anticodons, and that anticodon delivers a specific amino acid every time that codon is present in the mRNA, and that is how you decode the base code and turn that into a sequence of amino acids. Let's see. Yeah, and this just, again, reviews the same thing, right? These are your codons. Every time a specific codon shows up, there's going to be an anticodon that matches it, and that's going to deliver a specific amino acid. So every time there's UGG as part of your mRNA as a codon, then ACC tRNA anticodon will stick there and ensure that this amino acid, tryptophan, gets delivered. And then this, is that threonine? This yellow amino acid is going to get attached to the tryptophan. There are, and this is above and beyond what you need to know, some codons, like this one right here, for which there are no anticodons. So there are no tRNAs that have the anticodon UAG to stick to this and deliver another amino acid. And this is what terminates protein synthesis. When the ribosome gets to here, there's no tRNA to fill in the active site, and the ribosome just falls off of the RNA, and your protein is now complete. Again, it's mechanical. The ribosome knows where to start because of mechanical interactions with the mRNA, and the ribosome knows where to end because that's when the tRNAs stop showing up and it just falls off. And this just sort of sums it up again, everything we've already gone over. They're calling this the template strand because this is complementary to that, so TAC is complementary to AUG. And you don't need to know this, but it's fun to know. This strand here, which doesn't get transcribed, is called the coding strand because it matches the mRNA. So ATG on the coding strand is AUG in the mRNA. So this sequence matches this sequence just with the substitution of U everywhere there's a T up here. But this is how it works. It's all mechanical, straightforward. I said the ribosome knows where to hop on and knows where to hop off of the RNA, but it's not knowing. The ribosome fits at the beginning because of how the ribosome interacts with the beginning of your mRNA transcript, and it falls off at the end because the physical interactions that were keeping that whole structure together cease to happen when you get to that stop codon. That is it. I have more pictures here, but I don't have anything to say about them. They're just there because they are. So thanks for making it through this far. See you in the next screencast.